Well, no.
Yep.
All right.
How's it going?
Good. Happy New Year's.
Yeah, happy New Year's to you too.
Yeah. Yeah. As I emailed you, my brother was here, so
Yep.
it was nice to spend time with him. But like, I
Mhm.
just got nothing done. Obviously.
Hmm.
It just felt so unproductive like the whole week. But like, I needed the break.
Yeah, yeah.
So
Does this feel like your older brother, younger brother? Older.
younger.
Oh, yeah.
The older one I saw last week.
Got it.
But this week my younger came. Yeah. So
Nice.
had a good time. But
Awesome.
yep. But yeah, taking my polling tomorrow for Altius.
Yeah.
Um, so yeah, I mean, I haven't done that much this whole week, but like with the way we have our schedule set, I'm gonna take it tomorrow.
Yeah. You did
Um,
pretty well on the last one.
yeah, I was pretty happy with
Mhm.
how it came out. I mean, 507 after like couple bad, exams.
Yeah.
I'll take that. And
Yep.
also the Chem Phys. The fact that I got a 128 on this and a 129 on the AMC too. Like I'm very happy about CP.
Hmm.
Um, so as I told you, like the biochem, I'm gonna do a lot of UL for as well as section bank. When I was reviewing it, I mean, once again, this is just a constant thing that's happening is I'm making like these really bad mistakes. You don't understand. This was like really bad on biochem. There was a graph that said NO plaques. And so it was just like basically talking about the number of plaques. And it's like you add a drug and it was like Alzheimer's related, whatever. On the exam, I thought it meant no plaques. Like no plaques. So I literally got it wrong just because it said NO period plaques, which means number of plaques, but I thought it was just no plaques. And
Oh,
so you know what I mean?
yeah, yeah, yeah.
So instead of like an exponential up, I put the inverse one where it was going down.
Yeah.
So like I had the idea, I just literally, yeah, it was terrible.
Was there a period after the NO?
Yeah, there's a period after NO.
Got it.
Yeah. But what I'm also gonna try tomorrow is so I've been kind of doing the thing where I like do the discretes first. So I like bounce around and see the whole exam and I do discretes first.
Hmm.
But what I'm gonna try tomorrow is what like how people I guess normally take the exam. So I'm gonna try to just like start from passage one and just do it as it's supposed to be done instead of like doing discretes.
Yeah. If you do discretes first, you should have like the number, like the question numbers memorized so that you can just hit when you start the test, hit navigation, go to like question 14 or something, and then do those discretes. You know what I mean? So you have the
Yep.
knowledge of where the discretes are.
Yeah.
Okay.
Okay, anyway, so here, I will share the screen. Today I wanted to go over like 1 year old question and after that it was just a couple section bank like questions. So here, this is the first. Is it showing?
Hmm.
Okay. For this, like it's what is it called? The pseudo discrete sort of thing. Like we don't have to read the passage.
Okay.
It's basically something to do with like heat capacity and um yeah, bomb calorimetry.
Okay.
Water with an initial temperature of 20 Celsius is mixed with acetone in an insulated container. The temperature inside the container is monitored with a fiber optic temperature probe, and the mixture reaches thermal equilibrium at 30 Celsius. The capacity of the water is 420 joules per Celsius, and the heat capacity of the acetone is 150 joules per Celsius. What was the initial temperature of the acetone? So basically, like from what I remember you telling me, first of all, just like bomb calorimetry, I don't know if it has to do with this exactly, but like heat capacity is kind of the way we measure like how many calories is in something, right?
Well, heat capacity is how much heat can something absorb before it goes up in temperature by 1 degree Celsius.
And so what is this difference between heat capacity and specific heat? Because I confuse those two.
They're essentially the same. Specific heat just introduces a mass term. So, so like heat capacity is the amount of heat something can absorb before it goes up in 1 degree Celsius, and specific heat is how much like a gram of something can absorb, like how much heat a gram of something can absorb before it goes up 1 degree Celsius.
How much heat a gram can absorb before it goes up 1 Celsius?
Yeah. So
The
it's essentially just introduces a mass term in there.
and the other one is just how much heat it could absorb to go up 1 Celsius.
Hmm.
Okay.
Yep. So, you're probably thinking of this equation.
Right.
And this equation, this is going to be specific heat because we have a mass term in it, right?
Yes. Specific heat is MC, is what you're saying.
When you say MC?
Mass times heat capacity.
Oh, no, no. So this little c is the specific heat, which would be in joules per gram times degrees Celsius, which is why if you have a mass term in the equation, you would multiply that mass by the specific heat for the masses to cancel out, right?
Okay. So are you saying MC is heat capacity?
No, no, no. So C is specific heat, which has this as its unit. And so when you're dealing with specific heat, you use this equation because this equation has a mass term in it,
Okay.
right?
So, right.
So, but if we're dealing with just heat capacity, which is something that does not have a mass term in it, then your equation's just going to be like the heat capacity times the change in temperature without the mass term.
Mhm.
Okay.
All right.
So, yeah. So basically that. Let's see. Okay, so I guess this is mixing acetone with water. So water with an initial temperature of 20 degrees Celsius is mixed with acetone in what is that? All insulated container? Oh, in insulated and an insulated container. Okay.
Mhm.
The temperature inside the container is monitored with a fiber optic. So when you see fiber optic, think total internal reflection.
Mhm.
Um, and the mixture reaches thermal equilibrium at 30 degrees Celsius. That means that the water rises in temperature from 20 to 30. And the acetone probably starts off with a higher than 30 temperature because
Mhm.
it's gonna give off some heat to the water to make the water go up to 30, so acetone would go down to 30.
Yeah.
And then they give us the two different heat capacities. And then we just have to figure out the initial temperature of the acetone. Okay. So we can use this formula and use it for like the water, right? And also for the acetone. And if they're at thermal equilibrium, we can equate the Q terms, the heat terms. and then we can use that to solve for the delta T for the acetone.
Right, but so I mean, yeah, that part I kind of understood. My thing was just how I set it up in terms of the temperatures because basically like when you do set it up, you can set it up in a way where you get 58 or where you get 2, you know? So
So that'll depend on if you're using like simply delta T or if you expand that to mean final temperature minus initial,
right,
right?
exactly. Yeah.
So, okay, so let's see. So we can do heat capacity of water, and delta T for water is 10 degrees Celsius. Well, I guess I'll just put like delta T first for water, and that's equal to heat capacity of the acetone times the delta T for the acetone.
Mhm.
So now on the left here, this will be 420 joules per degree Celsius times the delta T is 10 degrees Celsius, right?
Yeah.
Equals the heat capacity of acetone, which is 150 joules per degrees Celsius. Now the delta T term we can solve for directly, and that'll be the difference, which means that whatever we get it for our delta T A here, we're gonna add that to like whatever we get at the end. Or you can expand it like this and solve for the T initial,
Yeah.
right? So whichever way you want to do it, I'll just do it with calculating the delta T. So okay, now we have basically 4,200 here divided by 150. One of these zeros leaves, 15 goes into 42. What, like maybe three times?
Mhm.
30 maybe?
Mhm.
Um, well, wait, a little less than 30, I guess. Um,
Yeah.
well, wait, what am I doing here? Okay,
That's right. I mean, 30 times 15 is 450. So yeah, a little less.
okay, a little less. So that's a little less than 30 is going to be our delta T. Um, well, okay, I guess then you can just add that to the 30 then, right? So you get, so 30 plus 30 is 60. It's going to be a little bit less.
Yeah, but so my question is this, like the it would be temperature final minus temperature minus
Mm-hmm.
hold on.
Initial. Yep.
So I was thinking about it this way. This is why I got it wrong.
Well,
I mean, like it would be
oh,
like let's say 28, for example, right?
huh?
So and then right, so I would be getting 2 here. Um, because I thought the final temperature for both was 30, you know what I mean?
Yeah, that's true. But we're
Right?
trying, yeah. So we're trying to find the initial.
Okay.
And so from here, you would just add, well, you could add the wait a sec, let me see. This will give us that, and because I'm distributing it, um, so then I would subtract 4,500 on both sides and then divide it by 150. So that seems like it's going to be more of a mess because you see I'm expanding the delta T to be, T final minus T initial, but then you take the heat capacity and you distribute it, right?
Yeah, I mean, I don't really understand what doing this like does. Because like I mean we're still gonna end up like it would still end up being probably something like this. Like 30 equals
Okay,
30
okay.
minus.
Yeah.
So
No, you're right. We don't have
um
we could just do the divide this on both sides, and then this, like, let's say like 28 is going to be equal to your final minus your initial. So to find so you can add the initial on both sides and then do the T final minus 28. Well, hold on. T final minus T initial is equal to 28.
so I'm saying, yeah, so
28.
final would be 30 technically.
Hmm. Something with the signs, probably.
Oh yeah, no, of course. That's like the whole catch. So
Yeah.
that's why I was just out. No, I was confused. That's why.
Uh-huh. Final. Wait, wait, wait. Oh, sorry. Okay. Yeah, I'm not sure what. Hmm.
Should I submit it?
Yeah, yeah, something to do with the signs here, just because we know that 30 minus 20. Well, okay, there's that, and then there's this, and then there's dividing it to get 28. And I think they just did the delta T part, and they used like logic after that because they have a plus there. Yeah.
Yeah. Okay, I mean, so technically, like logically, like I guess you just gotta know. I mean, it makes sense that like the it had to have been higher than 30. If
Yeah.
like
Yeah.
water was 20 and it went up, then the other thing has to go down.
Uh-huh.
So reach equilibrium.
Yeah. Yeah. The MCAT likes to do this type of thing where they where if you just plug in a formula, that's not going to get you the right answer. So you have to think about it like, logically. And logically, we know that the temperature of acetone must be greater than 30. So eliminating A would have been the first step. But I can see that a large percent, like, let me see that again. Yeah, 23% chose A because they just did the they just did the plug and chug uh
Hmm.
without really understanding it.
Hmm.
So, but yep.
This I got correct, but I just want to review it again
Oh yeah.
real quick. Um, this stuff, right? With like the critical angle and the total internal reflection.
Yeah.
Um, my question is in terms of like the critical angle versus total internal reflection. Like, Um, is this like I'm just trying to understand like when the incident angle is greater than internal angle, what like in terms of total internal reflection, does that just mean that there's like no refracted light?
Yeah. So basically, like the three main things that you need to know about this type of thing is that if you're going from if you're going from a less dense medium to a more dense medium, it will refract, it will bend towards the normal.
This is wait, so high to low is away, low to high. Right. So the more dense medium has a higher index of refraction.
Yeah. So if you're going from a lower index to a higher index, it will bend towards the normal.
Yep.
And that's pretty much it for that.
Yeah.
Now, when it comes to going from more dense to less dense, that's when other things can happen. So first, it will bend away from the normal rather than towards the normal. All right. Now, if you imagine this angle, the angle of incidence here, if you increase that, you're going to increase this angle of refraction.
Mhm.
Okay. So imagine, increasing this more and more until the angle of refraction here becomes 90 degrees.
Okay.
When your angle of refraction is 90 degrees, the angle of incidence is now called the critical angle. And that's when you have 100% refraction, like here, where it's completely along that interface.
Mhm.
Okay. Now, if you go beyond the critical angle, you can imagine the same kind of thing. Like you see how this is moving this way. So if it was here and this continued to, the angle of incidence continues to go beyond the critical angle, this starts to enter back into the original medium, the original denser medium. And that's when you have total internal reflection, where so the critical angle will produce 100% refraction. Beyond the critical angle will produce 100% reflection.
Got it.
And and that's how fiber optics work. So whenever you see fiber optics, let's think about this topic. Um,
Mhm.
Whenever you see, them even talking about refractive index, like anything like that, uh, think about this topic and the three main things. Three main things again are if you're going from less dense medium to a more dense, your light ray will refract towards the normal. That's one. Two, if you're going from more dense to less dense, it will refract away from the normal. Three, critical angle is the angle of incidence when you're going from more dense to less dense. That will produce a 90 degree angle of refraction. And then the last thing is if you go beyond that critical angle, you get total internal reflection.
Got it. Here I kind of eliminated A and B. I mean, yeah. Uh, Greater refractive index.
Mm-hmm.
So like when it's a greater refractive index, it's low to high, so it goes towards normal.
Mm-hmm.
This didn't really even ask this. It was just about velocity versus energy.
Well, they do say like the angle will increase, not change, decrease. That has to do with bending towards a normal, right?
Exactly. So I was between C and D.
Okay.
Um, I don't know why. For some reason I put C. But like yeah, I mean light just travels slower with you got to know that, right? It's in a more dense medium.
Let's think about what what is the index of refraction? It's the
Velocity. Yeah.
velocity, it's a speed of light in a vacuum, c, over velocity of the speed of light in the medium. And since this velocity must always be less than c, that means that the denominator must be smaller than the numerator, which means n must be greater than 1. That's why you
Mhm.
always see n values of like 1 point blah blah blah.
Mhm.
So yeah, this is the reason why it happens. Energy of light is not something you can really change, right?
Unless you change frequency.
Yeah, but even that doesn't really change, right? If you're
Mhm.
going from one medium to another medium, that frequency stays the same, right?
Yep.
Yeah.
Oh, here there, yeah, E equals HF. Okay.
Yep. And it says not the medium through which it travels.
Yeah. All right.
Hmm.
And this is the last zero problem. Here, it's like asking if you remove this resistor,
Okay.
how the current changes.
All right.
And my thought process, it was the wrong thought process, but I just always thought that like with current, if you have resistors that are parallel, like that the current through them is the same.
Um,
You know what I mean?
so let's think about this, right? When you have a junction, let's say this is I naught, it splits the current, right?
Yeah.
This can be I3 and this can be I2, right? And when they, come back here and here, they combine again to form the I naught, where I naught, oops, is equal to I2 plus I3. So whenever you have a junction, aka when you have things in parallel, like you must have a junction in that case. And the junction is where a current splits, right?
Okay.
So so yeah, you should have a different current go through resistor 2 and resistor 3.
But then when is the case that like when there's the same current?
When they are in series. Because if you think about it, right?
Oh, and it'll be different resistance when they're in series.
What's up?
Like the
Different
oh
voltage.
wait, no, voltage, right. Okay.
Yep.
Yeah, okay, got
So
it.
yeah. So if they're in series, they'll have the same current, different voltage. If they're in parallel, it'll be the same voltage, different current.
Hmm.
Yeah. So um and that's also how, something can like short circuit. So, okay, let's see. If all the resistors have the same value, um, so okay, if the resistors have the same value when they are in parallel, right? Then the current will actually be the same for both resistors. However, it will still be smaller than the original current. So, let me show you what I mean. So let's say resistor 1, resistor 2. Let's say that this is 1 ohm, and let's say this is 2 ohm, and let's say the initial like current is, I don't know, let's just say 5 amps. When it hits this junction, more of it will go through the resistor 1 because it has a lower resistance, right? And uh less of it will go through resistor 2 because it has a higher resistance. It'll go largely through the path of least resistance. So basically we can say like, it'll split from the 5, so the current will be still smaller than 5, but more of it will go through resistor 1 in this case. Now, if they're equal, if they're both 2 ohm, then the current that goes through resistor 1 is equal to the current that goes through resistor 2. However, both of these currents are still smaller than the original current, I not.
Mhm.
Okay.
Yeah.
So in this question, they are removing resistor 3. And when you remove resistor 3, which is in parallel, you get rid of this junction. It's no longer a junction. Then you just have resistor 1, resistor 2, resistor 4 in series. Right?
Yeah.
Wait a sec.
It decreases though.
Ah, Okay. Remember that resistors in parallel cause a smaller equivalent resistance. That's why you have the reciprocal sum.
Yeah. I mean, that's what I realized. That's probably what to do. Like,
Yeah.
so if you remove the resistor, then the resistance total is gonna go up.
Yep. Yep.
And that decreases I.
Yep.
Yeah, but like then when do I think about it in terms of like the equation versus I don't know, the equation versus like conceptually, if that makes sense. Because
So what I would do seeing this is I would do this. I would do first I would do like the equivalent resistance of these two in parallel. So that's 1 over Req is equal to 1 over R2 plus 1 over R3, right? And then I can add it in series. So R1 plus this Req up here plus R4. Okay. So that's me combining the parallel ones to get REQ and adding it in series to R1 and R4. And REQ must be less than R2 as well as R3, right?
okay,
So, but when I ultimately add them together like this, it's not a reciprocal sum anymore, it's just a regular sum, and that's why the resistance increases. So we basically, in removing resistor 3, we eliminate the parallel resistor, and parallel resistors cause the resistance to go down. So if we remove the parallel resistor, the resistance goes up.
hmm
Because if you have a junction, the current has a choice to go through another path. And if it can go through another path, right, it can, you know, go through something that is of a lower resistance.
okay,
Yeah, so see what they did? They added the resistor 2 and resistor 3 in parallel, right? And they got this. And that's also like kind of like a shortcut to like this formula.
yeah, yeah.
Yeah. Um, but yeah.
Okay, next. So, how do you like review with students these AMC stuff when because like sorry, Section Bank Volume 2 does not have the Jack Weston extension, right? So we can't like hide the answers. So do you just review it like this?
Wait,
I mean,
it doesn't let you.
no, like because I guess it's like yeah, like I just started doing it, but Like there's
What
no,
happens if you click that?
let me see. Yeah, like it doesn't
Oh,
work because it's new, and I guess Jack Weston didn't like update it yet, you
oh,
know?
got it, got it, got it.
Yeah.
So, but that question that you were showing me is a common thing that people, yeah.
Wait, what?
Oh, just the one that you were showing me about the
Which one?
organelles and nucleus.
This?
Oh no. Oh, yeah, this one.
Yeah, I mean this whole passage I kind of like
Uh-huh.
screwed up. So, but yeah, do you usually like before the Jack Weston extension existed, you just like I guess you just had the answers up. There's no other way to do it, right?
Yeah, I guess. Yeah. It's pretty annoying like why AMC does it this way. Like,
Yeah,
having it look nothing like,
the actual MCAT, yeah.
yeah.
Okay. Uh, yeah, I mean, I'd like to go through this whole passage so it makes sense to read it.
Yep. So, um, yeah. Uh, go for
Yeah.
it and talk about buzzwords as you see them.
Yeah. The ABO gene. And also as we go through it, I'd like for you to just like maybe critique my highlighting. I guess just like a minor thing, but the ABO gene encodes the enzyme glycosyltransferase, which catalyzes the final step in the synthesis of the A and B antigens on erythrocytes.
All right, what do you think a glycosyltransferase does?
Well, transferase transfers functional groups. It's a type of like one of the six classes, and then like GLY is like, I mean, has to do with sugars. Uh, Glycosyl, I mean, it's some sort of like sugar group
Mm-hmm.
that they're transferring.
Mm-hmm.
So, yeah, and
Antigen
antigen is it's like something that antibody binds to. I can't properly define it though.
does an antigen belong to a host cell, host or a pathogen?
Well, I feel like it could be either, no? Or I mean, I would say pathogen, but like there's two ways it could work. Like you can have the um like the macrophage takes in a pathogen, breaks it apart, and then presents that antigen on the APC
Uh-huh.
or wait, no, MHC, APC
Well, they both work. Because your APC is your antigen presenting.
cell, yeah,
So, you know, the antigen is still an antigen, which is something from the pathogen.
right. But the other way it works is if like sometimes it's an like endogenous thing. Like there's exogenous and endogenous.
Okay.
Exogenous is when it like it goes through phagocytosis, it brings in the pathogen, digests it, and then presents it. Or if the virus is replicating already for a long time, um, you can just present it because it's already endogenous.
Uh, yeah, I mean, I would say that a pathogen is should be considered like exogenous.
Mhm.
Um, but if you have like an autoimmune kind of reaction, that would be like uh antibodies kind of fighting against your own cells. But antigens should be stuff from the pathogen. So
So, but then why is like here it's A and B antigens? So my question is like, aren't A and B antigens like from your own blood or
yeah, yeah. So uh, you know. that can be like an autoimmune kind of thing. So like that's kind of like how blood type works. Like we have these kind of like carbohydrates on the surface of our blood cells that determine if it's like A or B or O or AB, right?
Mhm.
Um, because it would recognize other ones as being like foreign and treating it as if it's a pathogen. So yeah, so typically your antigens will be pathogens, but I guess in the case of blood type, it's not originally from a pathogen, but it can be treated as one.
All right.
So, what's erythrocyte?
Red blood cell.
Okay.
Polymorphism in the ABO gene are common and have led to the recognition of four blood types, O, A, B, and AB. Um, Polymorphism is what exactly? Just is it like multiple genes
Uh,
mutating?
it'll be so like typically you'll have like a SNP, a single nucleotide polymorphism. That's just something that's different in the genes. Like I guess in this case, like the polymorphisms in this ABO gene can lead to the different blood types. So they'll have like a different um like either nucleotide or number of nucleotides.
The glycosyltransferase responsible for the presence of the A antigen transfers N-acetyl D-galactosamine, where the glycosyltransferase that is responsible for the B antigen transfers D-galactose. Okay, wait. The glycosyltransferase is responsible for the presence of the A antigen. So N-acetyl D-galactosamine. Um,
Mhm.
all right. Well, it's like galactose, but it has an amine group, and on the amine, there's an acetyl group.
Mm-hmm.
Where the glycosyltransferase are responsible for the B antigen. Yeah, so it transfers the galactose.
Mm-hmm.
Okay, so just saying the differences between A versus B antigen
Mm-hmm.
and how it works with glycosyltransferase. Okay. The O blood group has a deletion near the N terminus of the transferase, resulting in a truncated protein in no antigen.
So that could be like perhaps like a nonsense mutation, like a premature stop codon. All right.
Yeah. Our proteins are created from N to C.
Uh, well, when you're combining them, you're going to be combining onto the C terminus with the N terminus of a new amino acid. But yeah, proteins will go from N to C. So, so like if you have an amino acid N to C and another amino acid N to C, the N of the second one binds to the C terminus of the first one.
Yeah. Okay. All right. Researchers investigate a relationship between ABO gene polymorphism and susceptibility to contracting malaria in the Brazilian Amazon region. Researchers use PCR and restriction enzymes for restriction fragment length polymorphism and analysis to assess the ABO genotype of each individual and enzyme length. Oh my God, it's a really long sentence. So, first of all, restriction fragment length polymorphism, when I see that, I know right away they're going to ask me about they're going to give me those nucleotides. They're going to be like, which one works with restriction enzymes?
You remember what term?
The palindrome,
Yep.
yeah. Um, yeah. So assess the ABO genotype of each individual and enzyme-linked sorbent assay ELISA and blood smear analysis to verify the presence of the protozoan Plasmodium falciparum, causative agent of malaria. Okay.
Yeah, I think I noticed from like, I mean, I haven't seen like that many of this new section bank, but so far I think I've seen like a couple of questions about, different types of uh single-celled organisms.
Okay.
Like, because typically I see like either prokaryote or eukaryote, but now, we're dealing with a protozoan here, which is a eukaryote.
Yeah. It's a single cell eukaryote.
Hmm.
Okay. Table one shows the relationship between the genotype and P. falciparum infection. So yeah, they're studying like if you have a certain ABO gene, like which one, depending on the one you have, like how to, relates to being infected by malaria, I guess.
Yeah, let's see. Alleles, phenotype, total number of tested individuals, and number of individuals infected by the plasmodium. So alleles AA, phenotype A, tested individuals 5, number of infected individuals 1, AO. The O is, I guess, recessive because you still get an A phenotype, a lot more tested individuals, and kind of like the same, perhaps a similar ratio. Um, like for the top one, it's 1/5, or which is like 20%, and this one's 21/46, which like it's a little bit more than 1/5. And then we have AB allele, phenotype AB, tested individuals, and then the ones who are infected, and then BB is B, and then that, and then BO, the O again is recessive, and then we get that, and then OO, we get that, and then we get a bunch of more people and all that stuff. Cool. Okay. So yeah, so this question asking us about, you know, the protozoan and, you know, I think you were thinking of it as a prokaryote.
Yeah, I mean, here they're expecting me to know that protozoan is a eukaryote then.
Yeah.
Okay.
Yeah. Um, so what separates a eukaryote from a prokaryote, typically people think is just the nucleus, but it's better stated as membrane-bound organelles, meaning eukaryotes have membrane-bound organelles, whereas prokaryotes do not.
Yeah.
So here they're talking about intracellular organelles.
Yeah. I mean,
Mhm.
definitely if I knew that it was a eukaryote, I wouldn't put A. Um, 'Cause, yeah, they just have DNA and floating around in
Yeah.
cytoplasm.
But I would say that since, even if you thought it was a prokaryote,
Okay.
um, well, actually, yeah, okay.
Yeah. Okay. Um, got it. Next is this, and I was like pretty surprised that this showed up.
Hmm.
Um, like what? They're telling me now. I guess I gotta know.
Yeah. Yeah, there's a
Between
lot
the
of weird
difference.
stuff that shows up in this section bank. I remember like there was a passage that I saw like asked about like the volumetric expansion. Oh, and then another one expected you to know that there's every 3.6 amino acids, there's a full turn in an alpha helix.
Wow.
Yeah, yeah.
Okay. Well, what I do know is that a light microscope is not as strong as an electron microscope or
Okay.
like electric microscope.
When
But
you
I
say
just
strong,
what?
you say strong.
Um, there's like a certain like when I say strong, I mean that the stronger ones can view smaller things.
Correct.
Yeah.
Hmm.
So yeah, I mean, I put C because I thought a protozoan is like smaller. I don't know,
Hmm.
but that's all I could really do.
Yeah. So, so this one, I guess, is something that you can just learn from this question. So we know that a light microscope, that's like the regular microscope that you use in like a lab, the regular one.
Yeah.
Um, and apparently the other ones, so the confocal one is for things that are smaller, like small molecules. Um, Scanning electron microscope, it, I believe it should be able to see things even smaller than the confocal, but it doesn't really tell you that. It just says that it's used to identify structures at the surface of cells. Uh, okay. Electron microscope, small portions of the cell. Yeah, these explanations kind of suck because like we would have to like look into this to be like, okay, like, you know, compare this small with, you know, like this small, like which is smaller, right?
Yeah.
So I would just like look that up. I would look up
Yeah.
the the stuff for each of these, yeah.
Yeah. And ask ChatGPT later to like differentiate between
Mhm.
them. Okay. Yeah. Next to this. So what describes the gene encoding the antigens?
Um, Gene encoding the antigens. Well, we know there's polymorphisms. But okay. Okay. Encoding a similar protein with different, so I don't know about the different functions in different species part. Um, but one gene with more than one allele occurring in a population at a rate of at least 1%. So down here they're saying like something about polymorphic. Okay. So this is just so it's the polymorphic part that this is asking us about, and apparently it means that each allele has to appear in at least 1% of the population. And then so now, okay, we can just add the rest of this stuff to like an Anki card. What's a pseudogene? What's an ortholog gene? What's a paralog gene? So,
I've never like really seen
yeah.
by the way, this is what I learned in my genetics class, but
Oh,
it's a 500 level genetics class.
yep.
Um, but I'm not gonna lie, I don't really remember it already, but um I just what I'm saying is like this is I don't know. I feel like pretty complicated genetic stuff that I have not seen anywhere else in MCAT, but
Yeah, like I
uh,
don't even, think that this is on the, the
I'll
content
learn.
thing.
Yeah. Okay.
But I would just make like an Anki card to be like, okay, what's a pseudogene? One silent gene that duplicated from another gene. What's an orthogene or ortholog gene? Uh, you know, yeah, I'm just making Anki cards for these.
All right. Yeah. So this I got wrong. Um,
Well, yeah, so this should be palindromic sequences, right?
yep. So I just want to explain my like train of thought. Um, so I like thought that this It's like really badly drawn, but like I thought that it would go like this. Wait, actually no. Okay, yeah, I mean,
So your palindromic sequence is going to be when the 5 to 3 gene
mhm.
is complementary to the 3 to 5 gene, which is the same thing going backwards, right? So, for instance, for this one, if I did 3 to 5 here, it would be C A A T T A G. And this does not show that it's complementary, right? Because you have A's, you know, and T's binding, supposedly binding to like these A's here are not going to hydrogen bond with these A's, and these T's are not going to hydrogen bond with the T's, right?
Okay.
Now, how about this one? 3 to 5. So it's C, T, 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 A, A, G. So you see how it backwards is complementary to it forwards, like the C hydrogen bonds with G, the T hydrogen bonds with A, right? So your palindromic sequence isn't just like a palindrome, it's when the 5 to 3 direction is complementary to the 3 to 5 direction. Because the 3 to 5 direction is just the 5 to 3 backwards, but it'll have to be complementary to it being backwards.
Wait, so the best way to find it is just you just write this backwards and just see if it matches up.
Yeah, so it must be so it forwards, think of that as the 5 to 3 direction. It backwards is the 3 to 5 direction, and it must be complementary to itself. Like the 5 to 3 direction must be complementary to the 3 to 5 direction.
Yeah. Oh yeah, as for the pattern of inheritance, that is most likely for the allele studied by the researchers. Um,
Okay? So the AB thing would be the codominance, right?
yeah,
And
the
for
AB
O,
phenotype you're saying.
mhm.
Yeah.
And for O, it must be recessive, right?
Yeah, because when it's AO, phenotype is A. Yeah.
hmm So there's no incomplete dominance because that's when you have like half of A and half of B, something like that. Or like, a white flower and a red flower and incomplete dominance is the pink flower.
Yeah.
So that's when you have like a mix of something in between the two. But we don't have that. We don't have like half of A and half of B. We have both A and B. So that's codominance rather than incomplete dominance. And we know that A is not recessive, so A is out, meaning choice A. And then D says complete dominance for A and O, recessive for B. We know B can't be recessive because, you know, we have B when you have B and O, right? And, that also means O can't be dominant because, you should see it in the phenotype then.
Was there any really way to like know if there's incomplete dominance though?
Uh, so like that's when you would have like a mix, like uh in between the two.
I get that, but like I'm saying from the like table, is there any way to determine incomplete dominance, you know?
Well, I guess like if it said something like, I mean, like no, but
Yeah.
it would say something like, you know, I don't know, like half of A, half of B, something like that. But I don't know how that would be depicted in the table like that. I'm gonna be right back. Let me just spell it. Have you ever had like a gin and tonic? Oh, you're muted.
It's crazy you say that because I had one it's my favorite like cocktail. I had it last night.
Yep. Now, have you ever seen what it looks like under a black light?
No.
So black light is essentially just like a UV light. It like fluoresces.
Mhm.
And it's because it has something, it has this here. Oh, wait. That looks bad. That's just because of the background being transparent, but
Mhm.
here's a one. It has this, right? And as you can see, there's lots of conjugation,
Mm.
right? And this is a medication used to treat malaria. That's why, like, the Brits, when they were like, carving up Africa, they created tonic water, which is basically quinine, which is this molecule here. And this is a typical like anti-malarial molecule. It's under the topic under the class of fluoro or quinolones, like fluoroquinolones or chloroquine. Um,
Mhm.
yeah. So yeah, next time you have, a gin and tonic, see if there's like a black light nearby and you can see that it will fluoresce, it will light up.
Okay.
Mhm. And yeah, they made the drink to treat malaria.
That's
And
crazy.
caught on from there. Yep.
The more you know.
Yeah, exactly.
While we're on that topic though, IR spectroscopy, there's vibration, while UV is absorption.
Um,
UV spectroscopy.
so I mean, there's gonna be like absorption and stuff, and oh, I see what you're saying. So like, yeah, so IR is basically like using a radio frequency to like shoot it at a molecule, get it to like rotate or vibrate or turn. Yeah.
Mhm. I had this. This was AMC. AMC.
Hmm.
Um, And so it was like absorption of ultraviolet light by organic molecules results in which process? Always results in what process? And the answer was excitation. And
Mm-hmm.
then C, it spoke about vibration of atoms and polar bonds.
Mm-hmm.
And they were like, this refers to IR.
Yep. So, so on one end, you can think about it as infrared, so it's not going to be using UV, but yeah, essentially what it does um, is like, yeah, it shoots like some infrared light at it, which gets kind of like absorbed, but as it gets absorbed, it like moves the molecule or rotates it or causes it to vibrate, and then it can shoot uh the light back, and then we get a measurement of that. Um, Whereas for UV vis, it's all about absorption and fluorescence because it absorbs the UV light and it can emit, as we know in fluorescence, it's about absorbing light that's a higher energy than emitting light. So UV is what you absorb, visible light is what you emit. So UV vis is a good way to check for conjugation, to check for triple bonds and double bonds and stuff. Yeah.
But when they say vibrate, what does that mean exactly? The IR?
Um, Basically that you can shoot some light at it and it will cause the molecule to well, when I say vibrate, I mean like it can either like kind of get stretched like left to right. It can turn or twist as well. So a lot of those things can happen. And basically it will shoot out some variation of the frequency that it initially absorbed um, for that. So basically think of it as like how should I put this? So you know how it's like okay, so if you had like this, it can like absorb it and kind of cause it to like stretch. Okay, so IR causes excitation of vibrations of covalent bonds within that molecule, and it only detects dipole bonds. And so you can have essentially like your um like I drew it here, but it disappeared. But let me draw it again. Yeah,
Where?
I'm just like drawing some stuff on the bottom left,
I can't see it.
just disappearing. But
Oh wait, I see it now.
okay, yeah, here it is. So yeah, it'll cause like this to kind of like stretch this way uh as it absorbs the IR radiation, and yeah, those vibrations of covalent bonds that are polar will have certain, depending on your functional group, will have certain, amounts of IR spectra that you get. Um, so that's why you would get like, yeah, something for like triple bonds, something for double bonds, something for single bonds. And, you know, double bonds will have less of a stretch, triple bonds will have even less, single bonds will have the most. So you get more of a stretch for like an OH group, for example. But yeah, causes um vibrations of covalent bonds within that molecule, and that's what IR spectra is, and it's used to detect functional groups, whereas UV is simply a measure of absorption, and that can tell you if something is, conjugated or has, triple bonds, double bonds, stuff like that.
Okay. All right. Then there's one other pass I wanted to do. I will be back in a second.
Yep.
Okay.
Yeah. So here, when they say a duct, they mean that something can, you know, be released or passed through, uh, not the circulatory system. So they're saying pancreatic ductal adenocarcinoma. We're talking about the pancreatic ducts. And we know that, when the acidic chyme in your stomach goes to your small intestine, your pancreas releases some bicarbonate ion, um, as well as, some enzymes and stuff which get released into the duodenum. So the insulin and glucagon is happening inside like the endocrine um, glands of the pancreas, but ducts are exocrine, meaning so like endocrine means it like goes into your blood, it gets like deposited in your bloodstream, but exocrine is like directly into something uh instead of through the bloodstream. So since they're saying pancreatic ductal, adenocarcinoma, we
Oh, oh, that
Mhm.
makes so much sense. Because yeah, I didn't even realize that. Because I was just looking at this, like, okay, it affects pancreatic tissue. So I just, yeah, I know it was between C and D because yeah, like the digestive enzymes are the exocrine pancreas. This is endocrine.
Mhm.
Um, but I wasn't sure which one it was. But yeah, now that I see the ductal,
Mhm.
that makes so much sense.
Mhm. Yep.
Yeah. Okay. Most of the absorption of nutrients is done by which part of the small intestine? The duodenum
Well, no.
or the jejunum and ileum?
Um. Oh, wait. The duodenum is where I think where most of the digestion happens.
Hmm.
And I know absorption of like water is in the large intestine, but let me see.
Yeah, water and salts is large intestine.
So primarily in the first two sections of the small intestine, so you can have in your duodenum, um, a good amount of like, I guess, iron, calcium, magnesium, and water-soluble vitamins. But in the middle section, the jejunum, that's where you have most of the nutrient absorption, such as carbohydrates, proteins, fats, vitamins, minerals, etc. And then the ileum, the final section, is specialized for bile salts, vitamin B12, and any remaining nutrients. So yeah, I would think about that as so the duodenum is where the most digestion happens, breaking things down, and a decent amount of nutrient absorption, but mostly like not like food. Like when I'm saying food, I mean like carbohydrates, proteins, fats. That
Yeah.
happens most. That's mostly absorbed in the middle part of the small intestine called the jejunum.
Okay.
Yeah.
Next is this. Which molecules transcribe GLUT5? It was just asking for,
Fructose, or
yeah,
so we know that it's not a disaccharide.
yeah, it's not.
But what would C be?
C would be sucrose.
Yep. Good.
Yeah.
And I guess B, yeah. Oh, and then
Glucose, and then
uh-huh.
the very bottom is maltose, right? Wait. I mean, it's either lactose or maltose, but
It might be just like two glucoses, maybe,
yeah,
which would just be like not something on its own, but like if
yeah.
you have a lot of it, you'll get like your glycogen and stuff. Um, oh wait, this is like a
Oh,
oh,
lactose.
lactose. So that's so yeah, I forgot that these are not the same, meaning this one goes up, this one goes down. So they're not the same. So one of them is glucose, one of them is galactose, right? Because lactose is a disaccharide of glucose and galactose.
Mhm.
Yeah. And fructose gives you this five-membered furanose ring because it's a ketohexose,
Okay.
right? Meaning, like, instead of the first carbon having the carbonyl group, oh, it just disappeared. But instead of the first carbon having the carbonyl group, it's the second carbon. And if since it's a second carbon, yeah, it's back. Since it's the second carbon, it's gonna close the ring like one carbon closer. So that's why you get a furanose instead of a pyranose.
Wait, it's gonna close
Mm-hmm.
like that.
Yeah, I'm probably wrong about these in the middle, but yeah, this will attack that. Whereas in a regular one, it would be like, you know, that, like for glucose.
Okay.
So, yep.
Might be what you're referring to.
Hmm?
This might be what you're referring to, the alpha helix thing.
Oh,
Or maybe not, maybe not, I don't know.
uh-huh.
But it has to do with alpha helix.
Segments of the GLUT5 primary structure correspond to a cytoplasmic protein region. Okay, let's see. Um, in the passage, like, or wait. GLUT5, a 501 amino acid transporter that contains intracellular N and C termini and 12 membrane spanning alpha helices. Typically it's in the intestinal epithelium, but it can be in these other things as well. Um, and they asked for in the question they asked for what, cytoplasmic or something? Yeah, cytoplasmic. So um something like this. Of course it disappeared, but
Yeah, so I know how to do it. I'm pretty
but
sure I have to press annotate. Okay.
we know that the okay. So your N terminus, your C terminus are cytoplasmic, and we have these 12 membrane spanning alpha helices. So now, okay, so like, let's say this is 1, 2, 3, 4, 5, 6. Half of these are cytoplasmic.
Okay.
I think, well, wait, from the 12, the answer is 7. So half of it, including, okay, half of it plus the either C or N terminus, right? Oh,
So
wait,
it's like
hold on. The N terminus is also cytoplasmic. Intracellular N and C termini, and then 12 membrane spanning alpha helices. So, so it's half of these and then the N one, but this is saying a cytoplasmic N domain followed by 12 transmembrane alpha helices. Therefore, there's a cytoplasmic protein region after every other transmembrane alpha helix. 1, 2, 3, 4, 5, 6. The final cytoplasmic region occurs after transmembrane alpha helix 12 and is the C terminus.
what?
I mean, and I don't know why they're not including that one. Considering the cytoplasmic regions that come after each alpha helix and the initial cytoplasmic region, there are a total of 7 cytoplasmic regions. Now, I understand from the 12 membrane spanning ones, half of that, and I understand like the C terminus, but why are they not including like the N terminus? Segments of the primary structure of GLET5, GLET5, intracellular N and C termini, and 12 membrane spanning alpha helices. So they're doing only what comes after the alpha helices. Maybe that has something to do with primary, because I mean, alpha helices are like secondary structures, and they're talking about the primary structure. For some reason, they're not including the N terminus here. Let me see the explanations for the other ones. You just scroll down on the right. Yeah. 12 transmembrane alpha helices, only 7, again, it's repeating that. 13 that are not transmembrane, however, only 7. Yeah, I don't know why they're saying 7, because we have the 6 from the alpha helices, the final one, which they're saying is the C terminus. So for some reason, they're not including the N terminus. Maybe, because they're saying that. Hmm, okay, maybe since the primary structure is the, N, your R group, your C terminus, and remember the new one
I found a picture of it.
binds to the C terminus. Oh yeah.
No, because I searched up this question like on the internet, and there was like already a Reddit thread,
Mm-hmm.
and they like linked this article too.
Yeah.
Um, one, yeah, 12, 12 membranes spanning out the helices. Wait, do you see it?
Yeah. So my guess is since they're talking about primary structure is the peptide bonds, and the peptide bonds are created like remember how I said like an amino acid is N terminus to C terminus?
Yeah.
So if you okay, here, what I wrote was back. So amino acid 1, amino acid 2, but you see that the peptide bond forms between the C terminus of that first amino acid and the N terminus of the next amino acid.
Yeah.
So if primary structure is the peptide bonds, then the N terminus, I guess, is not included in that peptide bond. So maybe that's why they don't count it. That's my guess.
Okay. So
One, two, three, four, five, six, seven.
there's 5 cytoplasmic loops. 5 cytoplasmic loops and the N and C terminus. So there's 7 total regions.
I guess this loop is on the outside.
But that's extracellular, yeah.
One, two, three, four, five, six, seven, and this one does not count.
I mean, I guess, but I
Yeah.
mean, the person that posted said that N and C counted, and then it was these 5. But like the one near the C terminal doesn't count. But
Oh, let's go back to that question because they did list the number. So, okay, 2, 4, 6, 8, 10, 12. Okay, go back to that drawing. Um, let's see. Wait. Um, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 2. So I guess like yeah, honestly, yeah, not sure
all
here.
right, it's fine. I mean, something that's unaffected is a negative control. Like there's no reaction.
So I asked Chat GPT and it told me that the N terminal is cytoplasmic, the C terminal is cytoplasmic, and then there's 5 cytoplasmic loops connecting specific transmembrane helices. And that's how you get your 7.
Okay.
1, 2, 3, 4, 5, 6. I think they're counting like an extra 1 in there.
Mhm.
Okay, let's see. Highly aggressive form of cancer, higher in individuals affected by diabetes. Okay. Higher levels. Okay, fructose entry into cancer cells is mediated by GLUT5 and using a PDAC mouse model to assess levels of GLUT5. They found higher levels in the PDAC affected tissue compared to unaffected pancreatic tissue. They then, used a GLUT5, transfected equal amounts of PDAC cells with either a GLUT5 overexpressing vector, GLUT5, or an empty vector in a sugar-free medium, fructose containing medium, or glucose containing medium. A cell assessed cell count after 24 hours, figure 1. So we see effective sugar content in growth medium and GLUT5 expression on PDAC cell count. Okay, relative cell number. That's going to be, I guess, the cancer cell, I guess. Oh no, yeah, PDAC cell. Okay, cancer cell. Okay, sugar-free, pretty much same. Fructose, um, in the empty vector compared to the one that is overexpressing GLUT5, we have a higher amount of the cancer cells. And then for glucose, there isn't a difference between the vector and GLUT5. Researchers also observed that when that compared to PDAC cells cultured in a sugar-free medium, PDAC cells cultured in a high fructose medium exhibited decreased aamc activity. aamc is a kinase that directly targets and inhibits mTORC1. mTORC1 inhibits proteins such as the autophagy inducer ULK1. So maybe I can say, GLUT5 proportional to PDAC and well, disappears, but let's see. Based on figure 1, what is the effect of fructose on PDAC cell proliferation? Fructose increases PDAC cell proliferation. Okay, so A says only in cells transfected with empty vector. that can't be true because we see that it's also, you know, increasing the PDAC cell number in the GLUT5 case.
Yeah.
And B should also be wrong for like the same reason, just the other way around. And then C, the same extent as glucose, which is what we see here compared to this. But only when GLUT5 is overexpressed. Hold on, I'm going to take a closer look at this. Star and pound compared to, okay, so the star is compared to the no sugar condition, the sugar-free condition, meaning the asterisks are statistically significant compared to that. But the pound symbol is comparing it to that of the fructose containing medium. So these pound symbols mean that they are statistically significant compared to the fructose condition, I guess. So to the same extent as glucose, only when GLUT5 is overexpressed, to the same extent as glucose, regardless of transfection with a GLUT5 expressing vector. Okay. Okay, well, it can't be regardless because this is a measurable, difference from when you just have the fructose.
Mhm.
So it does matter if you're using a vector compared to, the empty vector compared to GLUT5, right?
Mhm.
And this one is comparable to this. So it'll be the same extent as glucose, only when GLUT5 is overexpressed, meaning this gray bar is similar levels to this gray bar.
Okay.
Mhm.
Why is D wrong again?
Because D says to the same extent as same extent as glucose, regardless. So okay, fructose like increases this cell proliferation, the y-axis thing, uh, to the same extent as glucose, regardless of whether you're using a GLUT5 expressing vector here or if you have an empty vector here. So, if it's saying the same extent and we compared the empty vector, it's going to be this compared to this. And we can see that this fructose one is not the same level as this glucose one.
Okay.
Have an empty vector. When you have GLUT5 overexpressed, they do have pretty much similar levels. So
Okay.
the same extent only in the condition where GLUT5 is overexpressed.
Mhm. All right. The rest I understood. Um, yeah.
Okay.
So meeting on Saturday night.